two stages in meiosis, meiosis 1 and meiosis 2. Meiosis 1 is when chromosomes are replicated, crossed over, and reduced from diploid 2n to haploid 1n. Meiosis 1 is the stage where chromosomes are divided. For humans, 2n, the diploid stage, would be 46 chromosomes, and 1n, the haploid stage, would be 23 chromosomes. So one set, or n, is composed of 23 chromosomes. Crossing over occurs in the prophase of meiosis 1, but before this can occur, the interphase must take place. The nuclear membrane disappears, the spindle fibers form, chromosomes condense and duplicate, and homologous chromosomes pair to each other. They are connected by the synaptonemal complex. Now the crossing over will occur. A special enzyme unwinds the chromatids and reveals DNA strands. Genetic material is exchanged between homolog chromosomes. This creates new chromatids and variation in genes. The homologs pull away from each other. This ends prophase 1. At the end of meiosis 1, there should be two haploid cells with chromosomes with two sister chromatids. Meiosis 2 is similar to mitosis. At the end of meiosis 2, there are four haploid cells, each with two chromosomes with no sister chromatids. It is important to appreciate the significance of meiosis. In relation to natural selection, sexual reproduction shuffles the genes of successful individuals. The possibilities of combinations of genes are infinite. More importantly, meiosis allows reproduction to occur by reducing diploid cells to haploid cells, and because of meiosis, chromosome numbers of each species are held constant. There are many disorders caused by problems in the meiotic process like Down syndrome, Klinefelter syndrome, Turner syndrome, and Williams syndrome. Let's focus on Turner syndrome, caused by an abnormal count of sex chromosomes in the karyotype. Turner syndrome is a medical disorder that affects about 1 in every 2,500 girls. Most girls are born with two X chromosomes, but girls with Turner syndrome are born with only one X chromosome, or they are missing part of one X chromosome. The effects of the condition vary among girls with Turner syndrome. It depends on how much of the body cells are affected by the changes to the X chromosome. Girls affected with Turner syndrome are usually short in height, but growth hormones can help them grow taller. Turner syndrome prevents the ovaries from developing properly, which affects a girl's sexual development and the ability to have children. Because the ovaries are responsible for making the hormones that control breast growth and menstruation, most girls with Turner syndrome will not go through all the changes associated with puberty unless they get treated for the condition with growth hormones and estrogen hormones. Nearly all girls with Turner syndrome will be infertile. Turner syndrome may also be associated with kidney problems, high blood pressure, heart problems, overweightness, hearing difficulties, diabetes, and thyroid problems. Some girls with the condition may experience learning difficulties, particularly in math. Many have a difficult time with reading, map reading, or visual organization. Reproductive cloning is a technology used to generate an animal that has the same nuclear DNA as another animal. In a process called somatic cell nuclear transfer, or SCNT, scientists transfer genetic material from the nucleus of a donor cell to an egg whose nucleus and genetic material has been removed. The reconstructed egg containing the DNA from a donor cell must be treated with chemicals or electric currents to stimulate cell division. The cloned embryo reaches a suitable stage and is transferred to the uterus of a female host where it continues to develop until birth. Only the clone's chromosomal or nuclear DNA is the same as the donor.